Everybody. So I'm going to use your packets to actually start reviewing for the AP exam. And when you take the AP exam, it's going to be totally different than what we expected it to be in the beginning of the year. So I picked out some questions that I think will be really beneficial for you. And I'm going to go over how you answer these questions, and we'll compare what my answers are, based on the rubric from the College Board, compared to what you got. And you can see what kind of points that you're accumulating. And then I hope you can pick out your mistakes. You won't get the same exact questions, but you're going to have to answer questions that may involve many of these different things. So the idea is to really read everything carefully. So here's the first one. Table 1, effect of 0.1 millimoles of caffeine on memory in bees. So the first question they're going to give you is going to be some kind of scientific data and interpretation question. You may have to design an experiment. You may just have to simply interpret the data. They're going to give you 25 minutes to answer that question, so it'll be pretty lengthy. I do think it'll be somewhat similar to what you're seeing in the practice FRQs that you've been doing through AP Classroom. However, the answers may need to be a little bit longer and more detailed. For those details of how you're going to be answering the questions, I've uploaded a document for you, and that will also be an assignment this week for you to go through it and really take a look at what's there. Okay? All right, we'll get to that later. So here's table 1, and whenever you're given a table or a figure in a question, you should really spend some time taking a look at it. So I made some notes in here in different colors. Whatever is on the left side of the table is always going to be your independent variable. Whatever is in these parts over here on the right are your dependent variables. So for the independent variable, we have treatment, we have control, and we have caffeine. The control acts as the comparison group. This is your experimental variable right here. And then for memory, they're using the average probability of revisiting a nectar source for the bees. And you're looking at plus or minus 2 standard errors of the mean. Basically how far away you are from the mean. Okay, so for the control, we can look at time 10 minutes when they recorded it. It was 0.72 plus or minus 0.09, and then 24 hours later, 0.41 plus or minus 0.07. And then for caffeine, 0.83 plus or minus 0.07, and then 0.78 plus or minus 0.08. So the thing is to not necessarily really compare caffeine to caffeine 24 hours later. We want to kind of compare the differences between caffeine and the control, because caffeine is your experimental groups and you have 2 time points. So the question says, in flowering plants, pollination is a process that leads to the fertilization of an egg and the production of seeds. Some flowers attract pollinators, such as bees, using visual and chemical cues. When a bee visits a flower, in addition to transferring pollen, the bee can take nectar from the flower and use it to make honey for the colony. Nectar contains sugar, but certain plants also produce caffeine in the nectar. Caffeine is a bitter-tasting compound that can be toxic to insects at high concentrations. To investigate the role of caffeine in nectar, a group of researchers studied the effects of 0.1 millimoles of caffeine on bee behavior. The results of an experiment to test the effect of caffeine on bees' memory of the nectar source are shown in Table 1. And so I highlighted this part for you because this is actually what they're investigating, and so that's really what's telling you what the experimental variable is. All right, so some notes on the bottom. The larger the standard error of the mean, the more variation this is going to be in the sample. Okay, so it means you're getting further and further away from the mean on either side. All right, so for A, it says on the axis provided, construct an appropriate labeled graph. This would be a bar graph to illustrate the effect of caffeine on the probability of bees revisiting a nectar source, the memory. And so that's worth 3 points that are there. And so I'm going to come down and show you the graph. This is what it should look like. It has a title, It has the access labeled treatment, and here's the graph. Okay, so like in order to get all 3 points, you have to be scaled appropriately. So if you notice, I left 3 boxes here. I made each bar 3 boxes, left 3 in between. Here's 3 boxes. It's scaled appropriately. Okay, and the same for the sides. And so how do we know how to scale that? Well, it's just basic graph skills, right? We can see what the amounts, the average probability amounts are for revisiting the nectar source on their memory. And so we at least needed to go up to 0.9, based on the standard errors of the mean there. So that's down here. Okay, so this is totally fine to do. You can do that. And it's still appropriately scaled and fitting on the graph. And so here is our control, which is labeled. Here's the caffeine, control, and caffeine. Okay, so when you do the graph, they also wanted you to put in the standard errors of the means. And so how do you do that? Well, it's pretty easy. If we look at the control here, right? So here's roughly the 0.72 plus or minus 0.09. And so that's what I'm doing right here, right? So 0.09 is almost like a 0.1. So if this is where we're at here, and then we go 0.09 above and 0.09 below, roughly on that set 0.7 mark, then you've got your standard error bars, okay? And then if we look at the caffeine, that's here, we've got 0.83 and 0.07. And so we estimate those error bars just like that, right? So here's the 0.8, error bar in, error bar in. And then if we look at 24 hours, it's the same thing, right? So here's 24 hours, 0.83 and 0.07 and then 0.78. So we plot out our graph and then we include our plus or minus. And that's really how you do these. I don't think they're going to give you a graph to do. I think it would take too much time. What I think that they may give you, though, is a graph to interpret. And that's really what we're after here. So in order to see if there's a significant difference between the memory of bees at time 10 minutes, we can't say that there is, even though that this is lower, because whenever these standard error of the mean bars overlap each other, it means there's no significant difference between the two groups, okay? However, over here, you can see that spacing, right? And so we can interpret this a couple different ways. We can see that caffeine does have an effect, right? It has a significant effect after 24 hours on the memory. And for the control, like we can compare the control to the control over here. After 24 hours, the memory is much, much less. Not the case with caffeine, but we can say for certainty 24 hours later over here, there's a significant difference between the two groups, okay? So based on the results, describe the effect of the caffeine on the following, and that's really what you're doing with this graph. Okay, so for B, caffeine doesn't affect short-term memory at 10 minutes. Sorry, that got cut off. And then you also need to say that caffeine improves long-term memory at 24 hours. So that would be worth 2 points. Okay, so it's basically interpreting the differences between your bars here, right? Significant difference here, not so at 10 minutes. Okay, so that's worth 2 points. And then when you get to C, let's go read the question. It says, design an experiment using artificial flowers to investigate potential negative effects of increasing caffeine concentrations in nectar on the number of floral visits by bees. So you have to identify the null hypothesis. Always pay attention to what's being bolded there. You have to identify a control treatment and the predicted results that could be used to reject the null hypothesis. So for C, the null hypothesis, there will be no difference on the number of floral visits by bees if caffeine concentration in nectar is increased. So the control group, nectar without caffeine, and then the predicted results to reject the null hypothesis, the number of floral visits by bees must be significantly different at increasing caffeine concentrations. And that's all you had to do. So as long as you wrote a correct null hypothesis here, you identify the control, and you predicted the results, you get 3 points. If you didn't know how to do this, predicting results based on what's there, you don't get that point. Okay, but that's fairly easy to do, and I really think they're going to ask you to do something like this in a few weeks. We'll see. All right, and then for part D, researchers found that nectar with caffeine tends to have a lower sugar content than nectar without caffeine. Plants use less energy to produce the caffeine in nectar than they do to produce the sugar in nectar. Propose one benefit to plants that produce the nectar with caffeine and a lower sugar content, and propose one cost to the bees that visit these flowers that produce the nectar with caffeine and a lower sugar content. And so the answer to this over here is worth 2 points. So one benefit is that there will be more visits by pollinators, which means more pollen is transferred between the plants. Or you could have said plants have more energy available for other uses. So either one would garner a point. And then one cost to the bees is that they'll use more energy visiting more flowers. Or you could say they may produce less honey. Okay, so as long as you had one of these two here and one of these two here, you would get 2 points. Okay, so you want to stop what you're doing right now and add up your points and see what you got for this first question that's here. Okay, all right, so then we're going to move on. This is question 3 from the first part, and it's got really important information up top because it's telling you what this plant hormone does. Oftentimes on AP exams, I don't know if you've noticed on the FRQ, you may get examples of things that you've never heard of before. And it really doesn't matter because they'll tell you exactly what it is. Your job is to interpret it. This you should absolutely know how to use. All right, so gibberellin is the primary plant hormone that promotes stem elongation. So worthy of an underline. GA3 beta hydroxylase, right, there's its abbreviation, is the enzyme that catalyzes the reaction that converts a precursor of gibberellin to the active form of gibberellin. A mutation in the GA3H gene results in a short plant phenotype. When a pure breeding tall plant is crossed with a pure breeding short plant, all offspring in the F1 generation are tall, right? That means all the F1 generation are heterozygotes. So as soon as I read this sentence, I came right over here and let's take a look at what's going on, right? And so a pure breeding tall with a pure breeding short is going to produce 100% in the F1 generation that are heterozygotes, okay? And then the next sentence says when the F1 plants, right, these are the F1 plants, when they're crossed with each other over here, 75% of the plants in the F2 are tall and 25% of the plants are short. Right, and so this is our 3-to-1 ratio, basic Mendelian genetics here. We can see each of these has a dominant capital T, so there's your 75% and there's your 25% short. Okay, so you should be familiar with the mRNA chart that's here. For A, the wild type allele encodes a GA3H enzyme with alanine, a nonpolar amino acid, at position 229. The mutant allele encodes the GA3H enzyme with threonine, a polar amino acid, at position 229. Describe the effect of the mutation on the enzyme and provide reasoning to support how this mutation results in a short plant phenotype in homozygous recessive plants. Okay, so here's your A. It's going to be worth 2 points, and I'm just showing you because of the describe, and then you have to do this, right? You have to provide the reasoning behind it. Whenever I do any sort of AP questions myself, I'm doing exactly how I show you. I read the top, I think about what's there, I look at the chart, yep, it's the mRNA codon chart, I know I'm dealing with some type of protein synthesis, and then I go one at a time. So I'm only thinking about A right now. I'm not going to read B or C yet. I'm going to do A. Okay, so here's A. So you would be correct if you said the amino acid substitution changes the structure and function of the protein. The reasoning is that the mutation will decrease or stop gibberellin production. Okay, so you're describing the effect of the mutation, and then you're providing reasoning to support, right? So that would be worth 2 points here. Okay. For B, which was worth 1 point, using the codon chart provided, predict the change in the codon sequence that resulted in the substitution of alanine for threonine at position 229. Okay, so all we have to do basically is go up here, and here's threonine and here's alanine, right? How do we know? Because it says it right there. And so what do we see? What's different? We've got A's here and we've got G's there, right? So that's how we know. So a change from an A to a G in the first position of the codon would garner you that point. And then finally for C, describe how individuals with one heterozygous or two homozygous copies of the wild type allele can have the same phenotype. And basically, a heterozygote, right, it still has that dominant allele. And if it's two homozygous copies of the wild type, which is still dominant, there's enough enzyme gibberellin is produced from the dominant allele. And so we can see that right up here. Okay, so hopefully this little question that mixed a little bit of Mendelian genetics and protein synthesis made sense. So this would be what a part 2 question would be like when you are doing the AP exam. This would be one of like what they would consider to be a 4-point question or a 15-minute question to answer that. Okay? All right, so we're going to move on to 6. This is also like a 4-point question. And you've got a cell with DNA damage. It has an individual nucleus, right? They place it on a slide and they do gel electrophoresis. Okay, and so we're looking at intact DNA remains in the head and damaged DNA is in the tail. That's what it's showing you, right? So I just made a little note of that. Here's the intact DNA and here's the damaged DNA. All right, so the question says, a COMET assay is a technique used to determine the amount of double-stranded breaks in DNA, the DNA damage in the cells. The nucleus of an individual cell is placed on a microscope slide coated with an agarose gel. An electric current is applied to the gel that causes DNA to move, right? That's electrophoresis, which we just learned about, getting pulled to the positive pole because DNA is electronegative. And the DNA is stained with a fluorescent dye. When viewed using a microscope, undamaged DNA from the nucleus appears as a round shape, the head, and the fragments of damaged DNA extend out from the head. That's the tail. The length of the tail corresponds to the amount of the damage of the DNA. So they're telling us the longer the tail is, the more damage there is. All right, so you would get 2 points each here. You had to identify and provide reasonings to support how the property contributes to the movement during the COMET assay technique, right? So basically, they want you to explain gel electrophoresis. That's really what they want you to do. So for A, DNA has a negative charge. The electronegative phosphate groups on the backbone of the helix are pulled to the positive pole, separating the DNA fragments by size. Okay, and so you're identifying the property of DNA. That's here. And this is the reasoning that's there, and that's worth 2 points. And then for this question, in a different experiment, cells are treated with a chemical mutagen that causes only nucleotide substitutions in DNA. Predict the likely results of a COMET assay for this treatment. And so if you forget what a COMET assay is, you need to come back up here. COMET assay is the technique used to determine the amount of double-stranded breaks in the DNA or the DNA damage. So if it only causes a nucleotide substitution, then the tail will be shorter than a cell with double-stranded breaks in DNA, since the length of the tail corresponds to the amount of damage in the DNA, which they actually told you in the question. That's here. Okay, so you only have to do one thing there. That would be worth 1 point. So this is more of a 3-point question. Right, so that was 6. And then we'll take a look at 7. All right. Many species of bacteria grow in the mouths of animals and can form biofilms on teeth, the plaque. Within plaque, the outer layers contain high levels of oxygen, and the layers closest to the tooth contain low levels of oxygen. The surface of the tooth is covered in a hard layer of enamel, which can be dissolved under acidic conditions, right, or a lower pH. When the enamel breaks down, the bacteria and plaque can extract nutrients from the tooth and cause cavities. Certain types of bacteria, and they're telling you it's called Streptococcus mutans, thrive in the innermost anaerobic layers of the plaque and are associated with cavities. Other types of bacteria, like Streptococcus sanguinis, compete with S. mutans but are unable to thrive in the acidic environments. So for this question here, for A, identify the biochemical pathway S. mutans use for metabolizing sugar. And so we can identify that fairly easy because they gave us a pretty big hint there. It's anaerobic, so that's going to be glycolysis. You could have said anaerobic respiration as well, and you would get credit for that, right? And then how that pathway contributes to the low pH. Well, it's glycolysis, so S. mutans is going to metabolize the sugar to pyruvate. And when you're metabolizing to pyruvate, right, how it's going to contribute to lower pH is because glycolysis over time is releasing some extra hydrogen ions that are there, right? And so once it's metabolizing the sugar, then basically you're going to have a lower pH on the inner layers. All right, and then for B, normal toothbrushing effectively removes much of the plaque from the flat surfaces of teeth, but cannot reach the surfaces between the teeth. Many commercial toothpastes contain alkaline components which raise the pH of the mouth. Predict how the population sizes of S. mutans and S. sanguinis in the bacterial community in the plaque between the teeth are likely to change, right? And so they told you what happens in those conditions. So when toothpaste is used, which contain alkaline components, S. mutans is going to decrease because it thrives in acidic conditions, whereas the sanguinis will increase in population size because it hangs out in alkaline environments. Okay, so that's basically all you had to say here. All right, so this part, if you identified the process here, and then you explained why, this would give you 2 points, and then this was worth 1 point underneath. All right, so add up your points for that. Okay, and then the only other one I wanted to kind of go over with you was another one of the really long questions, which would be like the 25-minute question you may see for the first part of the AP exam. And so that's this question here, the cellular response to infection by pathogenic bacteria. And this is from 2018, actually. So here's number 2. So let's go over this one. And I always like to look at the diagram first to see what it's going to tell me, right? So automatically looking at this, I know that there's some cell response going on, and this is some type of cell communication based question, right? And it's telling us cellular response to the infection. And so we can see the numbers here. So we can see that here's the inactive caspase, but when the bacteria invades, it activates caspase, right? And this ends in an ASE, so we're dealing with an enzyme. So once this caspase is activated, there are 2 possible pathways for it. The first thing that it's going to do is it's going to take an interactive interleukin and make it active. And then we can see that that interleukin is going to go out through the cell membrane. And then the other pathway is that it takes gasdermin, and then gasdermin here creates, we can look and see, like there's a pore that opened up, right? So this goes in 2 spots and it opens a pore in the cell membrane there. What exactly gasdermin is or interleukin, who cares? It doesn't even matter what these are at this point for you or for us, but we can tell exactly what they do, right? And so I know this is being released from the cell, so it's got to be some kind of signaling ligand that the cell is infected, right? And this is putting holes in the cell. So this is, just by looking at this, caspase is in charge once it's activated of destroying the cell. It's the enzyme that causes cell death, okay? Or apoptosis from the beginning of the year. So let's look at the question. Some pathogenic bacteria enter cells, replicate, and spread to other cells, causing illness in the host organism. Host cells respond to these infections in a number of ways, one of which involves activating particular enzymatic pathways, figure 1 that we just went over. Cells normally produce a steady supply of inactive caspase 1 protein. In response to intracellular pathogens, the inactive caspase 1 is cleaved and forms an active caspase 1. That's step 1. Active caspase 1 can cleave to other proteins. When caspase 1 cleaves an interactive interleukin in step 2, the active portion of the interleukin is released from the cell. An interleukin is a signaling molecule that can activate the immune response. When caspase 1 cleaves gasdermin in step 3, right, here we are, the N-terminal portions of several gasdermin proteins associate in the cell membrane to form large nonspecific pores, right, large holes in the membrane. Researchers created the model in figure 1 using data from cell fractionation studies. So cell fractionation separates by density. In the experiments, various parts of the cell are separated into fractions by mechanical and chemical methods. Specific proteins known to be located in different parts of the cell were used as markers to determine the location of other proteins. The table below shows the presence of known proteins in specific cellular fractions. That's what's here. Okay, so you've got a Krebs cycle protein here, DNA polymerase, we've got glycolytic protein, a sodium potassium pump, and then this NF B immune response protein, right? And so in the whole cell, we can see that they're all present, right? However, fraction 1 is only where we see the Krebs cycle protein. When it fractionates again, right? So we're spinning this down into by density into smaller and smaller things. Then we can see that DNA polymerase is present here, but so is this NFKB, right? And then fraction 3, we've got the glycolytic protein and NFKB, and then fraction 4 is where you're dealing with the sodium potassium pump, right? And so the plus sign means that it's the presence of the protein in those fractionations. All right, so for A, you had to describe the effect of inhibiting step 3 on the formation of pores and on the release of interleukin. So you have to use the model and the description, right? So when you get a question like this, don't get overwhelmed. Just go back to the model, right? So if you're going to inhibit step 3, which means you're going to stop this, what is going to be the effect on the formation of pores and on the release of interleukin, right? So we can look at the model here. So if you're going to inhibit this, the pores aren't going to form. That's not going to affect this at all. It has nothing to do with active interleukin. Okay, so for A, by inhibiting step 3, gasdermin will not be cleaved into the N-terminal gasdermin, so pores will not form in the membrane. The active interleukin release will not be affected. So if you answered both of those correctly, you get a point here and you get a point there. Okay, for B, make a claim about how cleaving inactive caspase 1 results in the activation of caspase 1. A student claims that pre-infection production of inactive precursors shortens the response time of a cell to a bacterial infection. Provide one reason to support the claim. So they want you to do two things. Make a claim about how cleaving this results in the activation, and then provide a reason to support the student's claim. Okay, so if we come back up to the model, because that's really what you have to do, right? We're looking at taking inactive caspase 1 and making it active. Well, that requires bacterial infection. Okay, and then just if you're not good at reading the model here, you can come back to step 1. In response to intracellular pathogens, the inactive caspase 1 is cleaved and forms an active caspase, right? So a pathogen is disease-causing bacteria in this example. Okay, so for B, cleaving inactive caspase 1 results in the activation of active caspase 1 by changing the structure or the shape of the protein. You have to come up with some other claim that is different, right, from what's there. Okay, so and the reason for that the response time of a cell to a bacterial infection would decrease because cleaving a protein is faster than making a protein through protein synthesis upon infection. So that's, that reasoning, it's kind of difficult. This is a tough question, right? So we go back to enzyme activation, we can see what's here. And so like this, if we can deal with this here, that's certainly much quicker than having to go through protein synthesis, from the nucleus with mRNA doing transcription and translation out at the ribosome and all that travel time. Okay, so that would be for B. All right, for C, the student claims that the NFKB protein is located in the cytoplasm until the protein is needed for the transcription. Justify the student's claim with evidence. Identify 2 fractions where N-terminal gasdermin would be found in cells infected with pathogenic bacteria. So you need the chart to answer this question because you're dealing with the NFKB protein, right? So we have to come back to the chart, okay? And we can already see the 2 fractions, so you've got that answer right there, okay? So all you need to do is justify the student's claim with evidence. So for C, the NFKB proteins are found together. Right? Here they are. So they're found together in 2 fractions. So possible they're in the cytoplasm. And where they're located, it's in fractions 3 and 4. I'm sorry, it should say 2 and 3, not 3 and 4, right? We can see where they're located, okay? All right, and then for D, describe the most likely effect of gasdermin pore formation on water balance in the cell in a hypotonic environment. What a great question this is. You've got cell communication here, you've got enzymes working, you've got, you're dealing with protein synthesis, transcription, and translation. What a great question. And now we're dealing with cell conditions, okay? So just remember that when a cell is hypotonic, water is increased on the outside of the cell, right? So what's the most likely effect of gasdermin pore formation on water balance in a cell? Well, water is going to enter the cell in a hypotonic environment, right? Because if you have more pores that are forming here and the cell is in a hypotonic environment, then it's going to have more ways for water to get immediately into the cell, okay? And then finally for E, explain how gasdermin pore formation and interleukin release contribute to an organism's defense against a bacterial pathogen, right? And so the answer for this is actually in the reading, right? So you had to explain how pore formation and the interleukin release contribute to the organism's defense. So if you come back to the reading, it's telling you exactly what interleukin does. It's a signaling molecule that can activate an immune response, and it's telling you what gasdermin does, and it makes the large pores. And so for D, gasdermin pore formation will cause infected cells to lyse, right, or to break apart, and prevents bacteria from replicating. And then interleukin will act as a signaling molecule, a ligand, to stimulate the immune response to destroy infected cells. So you would get a point for either of those. Okay, so just remember for this one, you're dealing, this is my mistake here, I was probably answering too fast and not looking quickly, but you're at fractions 2 and 3 here, where those NFKB proteins are. Okay, all right, so great examples of questions on here. I'm hoping that this is helpful to you. And what I want you to do is to upload pictures of your answers with your point values and then total your point values underneath. I'd like to see how you did. Okay? All right.